Good evening, everybody. It's Doc Kyle, and tonight we're going to talk about the mechanism of action and clinical enzymology. So, remember that your enzymes are proteins and they have this 3D shape. And in this 3D shape, they make they usually make a cavity. And this cavity is lined by their amino acid residues. This specific amino acid residues, they hold a different charge. So they are making a specific combination based on the charge. So when your substrate comes along, a specific combination in your substrate based on the charge will be attracted to the active site, thereby leading to a binding. And this binding is not only quite strong, it's also quite specific. So after binding, the optimal side or the optimal 3D orientation of the substrate for chemical reaction is also achieved. No? So that's why we're getting a very fast and very very fast and very optimal na chemical reaction. That is your active site. Usually it doesn't have any water because if you add water in a chemical reaction manggod in this scale, it tends to decrease the rate of reaction unless water is needed, no, unless it's hydrolysis, no. But more often than not, the active site is a water free area. We also have what we call your allosteric site. It's another binding area in your enzyme that is not the active site, no. So a lot of things can bind there, including your inhibitors in your non-competitive inhibition. You can also activators can also bind there to activate the enzymes, because sometimes enzymes are not inherently active. They need an activator to activate them pa. So that's no. So I think that's all. So isoenzymes, no. So if enzyme A and B does the same chemical reaction and they basically look the same, but they differ in their genetic origin, they are what we call isoenzymes, no? So your isoforms naman, um, they they are basically the same enzymes from the genetic level and from translational. What happens is there are post-translational modifications. What does that mean? After they are translated from your by your ribosomes, no? Nasa like additional na characteristic yun na add. So that's why they're quite different, no? So kanya siya na bukol din ang pikas na bukol sa pikas, no? So that's basically they are the same enzymes. They are isoforms of one another. And in your case of isoenzymes, they are isoenzymes of one another. So we have this. So kani enzyme, no? And then sometimes your enzyme needs a cofactor. No, what does a cofactor mean? It just simply means that it aids, no, it enables the enzymes to produce your chemical reaction. The enzyme needs it to do its job. So sometimes these cofactors are what we call inorganic na mga molecules, no? And then we call them activators. Your inorganic molecules are your ions, your magnesium, chloride, no, selenium, molybdenum. If it's organic molecule like your nicotinamide diamine, no, nicotinamide adenine dinucleotide, NAD, no, or acetyl CoA, no, it's what we call coenzyme. So activator if it's inorganic, and then coenzyme if it's organic. Both are cofactors. Okay? So the enzyme part, the pure enzyme part, is what we call the apoenzyme. No? The protein lang dyan na part. And then the entire part is holoenzyme. So the cofactor simply means that it's a component that is non-protein. No? Non-protein ang cofactor. So it can be your activators or your coenzymes. So kato, the protein part is just the apoenzyme. The entire thing is what we call the whole enzyme. So, your coenzyme, biotin. So, usually they're vitamin precursors, meaning they're modified from vitamins. And these are the chemical reactions. So, we have another term, this zymogen, that we're... So, if we have an enzyme that breaks down proteins, like what we call protease, no? After translation ana sa imong ribosomes, pag active na siya dayon, patcho niyang cell, no? It will break down all the proteins inside the cell and then the cell will eventually die. So in order to combat that dilemma, these types of protein like your proteases are secreted as zymogens, meaning inactive. So in their amino acid sequence, there are additional non-functional. So kanitong functional, may additional non-functional na mga areas ay na mga amino acid. So because of the presence of these non-functional amino acids, the active site or the overall action of the enzymes is kulang, dili pa siya enough, dili siya mag-work pa. So eventually once it goes into its target location, which is usually the gastrointestinal tract, no, in which it will break down the protein intake, na enzyme mag-cut off ani na inactive part and then it's already active na. So the inactive enzyme, we call it zymogen or a proenzyme. So according to we have different types of nomenclature of our enzymes based on their action. No, according to the International Union of Biochemistry and Molecular Biology, no, old name ana is IUPAC. So International Union of Applied IU of Pure and Applied Chemistry, no. We have different naming. So the A suffix is derived from diastase. Kanitong gigamit para mo mag-rise ang bread, no. The first enzyme to be discovered by Emile Duclos. I don't know if that's the proper way to pronounce the name. So we have your one good way to have a mnemonic is OTHLIL, no? So we have your oxidoreductase from the name itself, oxidation reduction, no? So removal of hydrogen or basta oxidation reduction, diba? Or giving or taking hydrogen. Transferases, transfer of a group other than hydrogen from one substrate to another. Hydrolysis, usually it's breaking of bonds by inserting water to a bond. No? Breaking of bonds usually ng hydrolysis. Liases, no? Removal of groups without hydrolysis. Basta wala insertion of water, that's lias. No? Nalayas na siya. Isomerases, like I said earlier, we have different types. Your isomers, no? Isoforms, isoenzymes. Sometimes we conversion of the geometric, the 3D, or the positional isomers. Basta ilahi-ilahi niya itong arrangement sa enzyme. And then we have your ligases. Ang ligase is ligate, no, meaning joining of two substrate molecules. So kato ha. So this is just a short table. So we have different types of enzymes and there. What are the what are the clinical implications of enzymes? Simple lang kayo na most of your enzymes are intracytoplasmic, meaning intracellular sila. Kung na-damage ato na tissue kanang example, nag-heart attack ka, na angol imuhang heart muscle, mag-release to na enzymes and then they will increase in your blood. If we detect those increase in enzymes in your blood, then we can safely correlate that with the clinical. For example, sakit yung dughan. So, nag-check ta sa iyahang cardiac enzymes. Then, nakita natin itaas ulit yung cardiac enzymes. So, most probably na siya heart attack, no? Heart problem. So, kana same sa imong pancreas, sa imong pancreas, sa imong liver, so na siya. So, increases of these enzymes, like example, this creatine kinase, no? specific. It's quite specific. Example, the CKMB isoenzyme, isoform rather. It's in myocardial infarction, heart attack, no? So kana, kung mataas na sila na enzyme, most probably na problem dito na area kay there is the only way for the enzymes to escape intracellular from the intracellular space is for the tissue to number one, necrosis, buto siya, maguba, release ang enzymes, or there is damage to the cell, meaning there will be leaky cytoplasmic membrane, mag-leak siya outward, no? Napod sa malignancy, kung nakay-cancer, what will happen is the cells will um, continually undergo mitosis. If there is lots of cellular division, then there is also lots of cellular death. So kung may death, may release of enzymes. No? So the enzymes usually is what we call kanang cancer antigens or cancer markers if in the context of malignancy. No? So that's the purpose of these enzymes. I think we will talk about this more in clinical chemistry. So the allosteric site, like I said earlier, it's the other binding site. So it can be either be inhibition, so it can either be activate or spot. No? So I think so. The two conformations of enzyme is the active, it's the relax, no, and the inactive, also known as stout, no. So I think that's self-explanatory, but ngano siya inana, no? So if yung active site, kanina lang ako ang kanang analogy. So think of the active site as always yearning for something to be inside it. That's the only way na ma-relax siya, no? Because you really need to, this active site, it needs energy. It needs energy to, not necessarily energy in the context of ATP, but there's tension ba to hold the form. But once a substrate goes in, kato electrostatic bonding, ma-relax na siya kay ang tension ma-relieve na siya. So that's why it's the relaxed form that tightly binds the substrate. And it's the inactive kato siya, relaxed conformation binds tightly. And inactive, also known as a taut, which binds the substrate less tightly. So the nature of the active site, like I said earlier, it's a combination of your combination of charges from your amino acid residues. So sometimes there is what we call a nucleophile, no, an electron-rich substance that tends to react with sites of positive charge or polarization. So kani siya combination niya sila sa nucleophile o electrophile within the active site. So depende lang po na sa substrate. This ensures the optimal orientation and the binding of the substrate, no, for efficient chemical reaction. And then the electrophile is an electron-poor substance that tends to react with negative charge or polarization. So kana siya. So there are factors that influence enzymatic reactions. So same lang yapon before. So your substrate concentration, that is your first order kinetics, no, substrate. And then enzyme is zero order kinetics, also known with your Vmax. Enzyme concentration is the basis for your Vmax, no, pH. So dependent on pH because like I said, ang amino acid residues, they are dependent on the binding sa pH because we have what we call acidic and alkaline na mga amino acids, diba? So they work well depending on the pH, no, of the solution. So some enzymes work well with your kanang acid na environment, no. So kato temperature, like I said earlier, mag-increase ang temperature mag-increase ang reaction if increase yung temperature until eventually ma-denature siya. Especially pag 40 to 50 degrees na ka. Presence of cofactors and coenzymes. Kung wala kay cofactors and coenzymes, dili mag-proceed ang imong reaction. Okay. And then if you have inhibitors, I think that's self-explanatory. So that's all. It's quite a short topic. So if you have any questions, feel free to PM me if you have any clarification. So thank you guys. Bye bye.